jumping right back into the leukocytes, aka the white blood cells. Please, 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 please know your terminology. Again, this is part 2 of blood, of biology of cells and tissues. Again, if you're watching this video, if you haven't watched part 1, jump back, watch part 1, and then come back to this one. And as always, leave a like, subscribe to the channel if you haven't already, as it really helps me out. And if you have any questions, comments, concerns, leave a comment, and I will get to it as soon as I can. So general structure and functional characteristics of our leukocytes. So these are white blood cells. Again, these are our only complete cells, which means they have the nucleus, they have the nucleolus, they have all those organelles. You know, they can do all of the stuff that cells do that we've talked about up to this point. They make up about 1% of our total blood volume, and the normal range is between 4,800 and 10,800, about 4,000 to 11,000 per microliter of blood. Okay? Make sure we know our normals. Again, throw in there 4,000 to 11,000 is the typical normal range. And these are for our defense against disease, our immune system. Key points that we're going to go over. So they leave the capillaries via a process called diapedesis. Practice saying this word a couple of times because it's going to come back a lot in the future. You need to make sure that you know this word. And they move through tissue spaces by what's called amoeboid motion. If you don't know what an amoeba is kind of like a single-celled organism. It kind of looks like a little blob, and it moves around through stuff. As well as chemotaxis, or chemotaxis, depending on how you want to say it. Okay, so these 3 terms we're going to dive into. I have some more definition information stuff down here about all of that, but it's also going to be on the future slides. So diapedesis, it's the process by which white blood cells move out of the bloodstream. You don't need to know all of the nitty-gritty details about this, just that it's this process that the white blood cell goes from floating and moving along through the blood vessel to getting out through the wall of the vessels, specifically the capillaries, into the surrounding tissues. Okay? That is what we need to know about diapedesis at this point. In the future, we'll learn all of the different components of it, but for now, just this. I'm going to highlight. That's what we need to know right now. Now the little picture, just kind of all this going through. Amoeboid motion. So amoeboid motion refers to the type of movement the white blood cells do to move through tissues. So the way that amoeboid motion works is that basically they create these little extensions off. These are called pseudopodia, or fake feet. Podia, podiatry, pod means foot. Pseudo is fake or synthetic. So a fake foot in order to crawl through tissue spaces. They kind of shoot out a fake foot and it almost like pulls them along. I threw in a little YouTube video here as well. Let's see if it'll pull up quick, just so you can kind of actually see this in action. You can kind of see how the legs, these little false feet, extend out and kind of pulls it along. This is what amoeboid movement looks like. You don't need to know all the, again, you don't need to know all the details. I'm just, I find it's much easier to be able to understand something and be able to answer questions about something if we know what it actually is instead of just knowing the word. Finally, chemotaxis. This is probably one of the least explained terms in science courses. I remember I've heard this term 100 different times, but I feel like nobody really explained it super well. So chemotaxis is the directed movement of cells towards or away from a chemical signal. Most commonly, it is towards. When we talk about, cell injury, when we're talking about white blood cells, I'm talking about cell injury, cell death, cells tend to move, the white blood cells need to move towards the site of injury. So whenever a cell gets injured or damaged, it releases these chemical signals called chemokines or cytokines. These are two of the big chemicals that they can release. And essentially, it's like a call. It's like a message. It's like a cry for help. The chemical message gets sent out, and the white blood cells pick up on that, and then they go towards that location. Okay. I mean, this can also be, not just chemokines or cytokines, but other non-body cells, so like bacteria, can also give off certain signals that then cause chemotaxis. So this is an example. This is a neutrophil actually chasing a bacteria because the bacteria is giving off a different chemical signal, and the neutrophil picks up on that, and this is chemotaxis, the movement of these white blood cells through the tissues following these chemical signals. So kind of chemotaxis, diapedesis, they both go hand in hand. And this, you can also kind of see the amoeboid motion with this. See how we kind of get these little false feet out here moving around, how it kind of like pushes out. So there's a good video kind of all of those. So that is chemotaxis, the movement, that guidance system, that direction towards the injured tissues or the pathogens, whatever needs to be worked on. Here's another little video for you guys. If you have my notes, make sure, like, this is another really good one. It kind of goes a little bit more detail, but it kind of shows chemotaxis, it shows diapedesis. It's really easy to watch. So now we know that we have our normal ranges, and so we know if we're outside of our normal ranges, that typically indicates some kind of pathology. And when we have high amounts of white blood cells, hopefully we can correlate this to some kind of disease, kind of sickness. The term that we use for having high numbers of white blood cells is leukocytosis. Leukocytosis, so leukocytosis. Leukocytosis, so white blood cell osis. I don't remember what the suffix. Let me look at that up real quick. Osis is a process or condition, so it is a white blood cell process. So we are creating more and more white blood cells. Blood cells are up because of some kind of condition. Again, about between 4 and 11,000. Anything greater than 11,000 is leukocytosis. It's a response primarily to infection is the big one, but also things like inflammation, stress, tissue damage, anything like that as well causes an increase in white blood cell count. Getting a little bit deeper into leukocytes, like what they are, what they're called, there are two groups of, or I should say two categories of white blood cells. We have, and they're based off of what they look like when we stain them. We have granulocytes, which have visible cytoplasmic granules when we stain them, and then agranulocytes, which do not have those cytoplasmic granules. And we're going to look at these all a little bit deeper. We're going to come back to some of these little things. This is just little mnemonics and stuff to help us remember. We're going to talk about all of these right now. But this is mnemonic to remember the decreasing abundance in the blood. Never let monkeys eat bananas. So in the order, neutrophils, which are granulocyte, lymphocytes, which is an agranulocyte, monocytes, which is an agranulocyte, eosinophils, which are granulocytes, and then basophils, which are granulocytes. Again, make sure that you familiarize yourself with this terminology, especially eosinophils, because this is going to come back next trimester in Biochem 2. We're going to really dive into kind of eosinophils, how they're produced, kind of what goes on, all that whole sort of category. And we need to have a solid foundation in order for that to make sense next trimester. Again, so here is just our order. So remember, never let monkeys eat bananas. How much are in each of those? So splitting these up, granulocytes versus agranulocytes. Here we can see we have the neutrophil, eosinophil, basophil. We're going to start by talking about those granulocytes. So these are the most numerous, so I should say granulocytes, starting with neutrophils, okay? When we talk about these, we're going to want to remember, you don't have to remember the percentages. You might end up with a question or two about the percentages. They're not super important for long term, so I wouldn't worry too much about those, but if you want to remember percentages, you can. So these are the most numerous white blood cells. They're twice the size of red blood cells. The granules, they have those. They contain proteins, specifically these proteins called defensins and lysozymes. Remember lysozymes? That sounds familiar to like those lysosomes within cells. That's for phagocytosis, digesting stuff, breaking stuff down. That's what the neutrophils' jobs are. Some of the naming stuff, this is another, vocab that you need to know. You'll see this term polymorphonuclear leukocytes, because the nucleus has like little lobes to it. Polymorphonuclear. It's just a word you need to remember. There's most of the time in this upcoming test, you will see a question that's something along the lines of which of the following is another name for neutrophils. Or it'll describe something about neutrophils, saying the most numerous white blood cell is, and this will be your answer choice, the polymorphonuclear leukocyte, okay? Or the polys. Something like that is almost always a test question. Neutrophils, their main job, when we see them really active, is bacterial infections, but they're also kind of the general purpose white blood cell. They deal with it all. Here's our polymorphonuclear neutrophils. See all of those lobes, lobule right there, lobes, all of those. That's the dead giveaway that it is a neutrophil. This is also key when we're asked to identify these on lab exams. We want to make sure we see all the different lobes. Next granulocyte, eosinophils. This is a much lower amount, 2 to 4% of all leukocytes. They have 2 lobes, so they kind of look like earmuffs. And these contain digestive enzymes. And specifically, these do 2 jobs. They deal with parasites and they deal with allergies. That's what we want to remember about eosinophils. 2 lobes. They deal with parasites and allergies. 2 to 4% of all of our leukocytes. Those 2 lobes, easiest way to recognize them are the 2 lobes and then the darker, they call it red, but darker granules around the nucleus. Okay, the two-lobed nucleus. Here, you picture more of some of these white blood cells. This right there is our eosinophil. I took my best guess of which ones these are. I don't remember. She never actually went specifically what these ones are. This one's either a monocyte or a neutrophil. I'm not sure which. This one isn't a great looking one, but I believe it's a lymphocyte. All of these guys floating around, these are our red blood cells. And then these little purple dots in here, I'll add these little guys right in there. Those are our platelets floating around. Again, I'm not 100% confident on that just because it looks multilobular, but it also kind of has the shape of a monocyte, which we'll look at later. Okay, moving on. Basophils. Basophils, remember, these are our least common. Never let monkeys eat bananas. Bananas being the least. So this is 0.5 to 1% of all of our leukocytes. They are bilobed nucleus, obscured by basophilic specific granules. These granules contain these two chemicals, heparin and histamine. So probably a good idea, if we drop back really quick, make a chart with some of these. Make sure I don't think I put a chart in here. Make a chart that contains the following. Or heck, let me do it for you. We're going to add a new slide here at the very bottom. Okay. So we're going to have, here, I'm actually going to do this on a different, I'm going to start the chart for everybody and I want you guys to finish it. So we talk about neutrophils. Let's look and see kind of what things we want to add in here. So we have neutrophils, eosinophils, and we have basophils. So neutrophils, 50 to 70%. So what other important things they have? Some of the, if we mentioned the proteins, defensins, lysozymes, important info, the recognizing it, how to recognize this one has multiple lobes for nucleus, polymorphonuclear. Bam, that's our granulocytes. Bacterial infection. Eosinophils, we got 2 to 4%. And it has digestive enzymes, parasites, and allergies. What's its, how to recognize it? Has 2 lobes. And then bright red granules. Fine, we got basophils. Basophils is 0.5 to 1%. Heparin and histamine. These are our these are inflammatory chemicals, specifically a lot with like immune type reactions. I think allergic, so allergies are a big one, and then pathogen defense. Best way to recognize it, this is just, you can't even see the nucleus. Can't see the nucleus because of all the granules. There we have a bunch of stuff right there. Let's add this. There we go. This is what we want to, how we want to break this down. Take the most important information, throw it in here so we can review it. Agranulocytes. I'll leave you guys to do the one for agranulocytes. So agranulocytes, they lack those granules. There's two types. We have lymphocytes and we have monocytes. Both have either spherical or kidney-shaped nuclei. So lymphocytes, again remember, never let L. The second most numerous white blood cell. That accounts for 25% of our white blood cells. There's a large dark purple circle nuclei with a thin rim of blue cytoplasm, mostly found in the lymphoid tissues. And it breaks it down into two other types, T lymphocytes and B lymphocytes. T lymphocytes are virus-infected cells and tumor cells. You'll see these come up a lot in future courses because the immune system is very, very important. And we learn a lot about it. Then the B lymphocytes give rise to plasma cells, which produce antibodies. So T lymphocytes, virus infection and tumors. B lymphocytes are our antibodies. Again, the antibodies also respond to viruses. So if we want to create something similar to this, lymphocytes, there are 25%. What it contains, doesn't give us any of the specifics, but we know its job. We have T lymphocytes are virus and tumor. B lymphocytes are our antibodies. How to recognize big nucleus. Right there. Started it out for you. You guys can finish it. Here are lymphocytes right there. Lymphocyte. Very dark nucleus with thin line of blue all the way around right there. Clinical application, or also known as our pathology, because of their central role to immunity. Lymphocytes are important in many diseases. There is a condition called a lymphoma. This is a cancer. You'll learn lots and lots about lymphomas in the future. But these are essentially when we get a cancer of our white blood cells. So the white blood cells that are dividing fail to undergo apoptosis when they are not up to snuff. So the white blood cells that should, recognize that there's an issue with them, and then they should just get broken down, don't do that. So these are often considered malignant because they can spread to a bunch of different parts of the body. So basically the white blood cells, because some white blood cells can last and live for years and years and years, they can travel to different parts of the body, get into other organs, get into other systems, and then just sit there and continue to grow and proliferate and all that kind of stuff. And it gets bigger, 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 and causes a lot of mayhem in the other organ systems. Next, we talk about monocytes. Monocytes are the largest of all the leukocytes, 3 to 8%. What's our mnemonic? Never let monkeys eat bananas. It's 3 to 8% of all of our white blood cells. Dark purple stain, we have a U or kidney-shaped nucleus. These ones are the ones that leave circulating. When we talk about diapedesis, these are the guys that we're talking about. These monocytes are big ones that are doing that diapedesis. They leave the circulation into tissues and differentiate into what are called macrophages. And we've talked about macrophages a little bit before. So monocytes turn into our macrophages once they undergo diapedesis. There are our monocytes, kind of the kidney bean or U-shaped nucleus right there. This last little picture here this is a really, really good picture to study. Kind of look at each of these and compare them, contrast them. Multiple lobes, earmuffs with the red, super, lots and lots of granules, mostly just nucleus, and then U or kidney shaped. Last one that we have to talk about are our platelets or our thrombocytes. So platelets are actually fragments, they're cell fragments of a much larger cell that is called a megakaryocyte. This is just more word definition that we want to know. Question that you could get asked is something along the lines of platelets are fragments of which of the following? Or platelets are derived from or created from which of the following? Answer being the megakaryocyte. These ones, they have a whole bunch of different chemicals to help the clotting process. Remember that platelets are with clotting, and the normal range are typically 150,000 to 400,000 platelets per milliliter of blood. There we have our little platelets. I pointed one out earlier. And there we have it. That is all of our material on the blood. Hope you guys enjoyed. Again, any questions, comments, concerns, please drop it down. And again, if you don't have my notes or don't have access to them and you will want them, please feel free to reach out. There's a link in the description. It takes you straight to a Google form that you can fill out and go through it. And it will tell you exactly how to get access to all of these. Thank you and have a good day and good luck studying.